He earned his PhD from the National University of Singapore, where he conducted extensive research in microbial genomics, contributing valuable insights into the genetic diversity and evolutionary dynamics of microorganisms. With several years of experience in bioinformatics, statistical analysis, and computational biology, Dr. Mukherjee brings a strong interdisciplinary approach to addressing complex biological questions. Beyond his scientific accomplishments, he is deeply committed to mentoring students and young researchers, actively contributing to capacity building and skill development in the field of genomic sciences. Please join me in warmly welcoming Dr. Arijit Mukherjee.
Right, that was a different kind of introduction. All right, so I will not introduce myself, and I'll just delve into the topic then. You can share my see my screen, right? You just okay. Is my screen visible? And I can go on?
Yes, sir. It's visible.
All right.
Oh.
Yeah, because I can't see you. You have to be vocal. All right. I'll be talking about my PhD work because I have recently joined my postdoc here, and mainly sharing on plants and the microbes. So I work more on the plant-microbe interactions, and the new concept that we brought in between plants and the microbes is the trade-off between plants and microbes, interorganism trade-off. So I'll be talking about that, how we discovered this phenomenon, and what are the techniques and how we approached it, right? So first, a little bit of introduction, because I know you're coming from diverse backgrounds. And then what is the question that I tried to address during this work, my findings, and summary. All right, I work with the plant microbiome. So like the humans, the plants are also colonized by different microorganisms. And this can range from protists, archaea, viruses, bacteria, and fungi. And they are everywhere on the plant parts, be it in the rhizosphere, on the root region, inside the roots, which is called the endosphere, and even on the leaves and inside the leaves, or even the seeds, right? So plants are colonized by these microorganisms, which are numerous and diverse, which we call microbiome. And one thing I would like to emphasize is that this assemblage between the plants and the microbes is at least 450 million years old. So they have been interacting with each other for millions of years, right? So they have evolved some functions. And one of the functions that I focus on is how the microbiome influence plant nutrition, because plants will require elemental nutrients to complete their life cycle, right? For example, I mean, what I'm showing here is one of the examples where the rhizosphere microbiome, or the microbiome that resides in around the plant roots, and how they help plants against nitrogen deficient conditions. So the bacteria residing plant roots can help them acquire nitrogen when nitrogen is less in the soil. Similarly, for phosphorus, when plants are encountering phosphate stress, the microbiome help them to cope with that stress by downregulating the immune system or defense system of the plants, right? So this kind of balance is achieved by the microbes that are residing in the roots. In the case of sulfur, what people know is bacteria can also produce volatile compounds. Here I'm giving an example of dimethyl disulfide, which is DMDS, and this volatile can be taken up by the plants, and that can be integrated into plant sulfur nutrition to alleviate sulfur deficiency situation in plants, right? So these are the knowledges that we know in case of plant nutrition mediated by sulfur. What we also know is that plants and the microbes also communicate via a lot of chemicals, right? The microbes can also secrete chemicals, and the plants can also secrete chemicals. For example, I'm just giving an example. Plants can produce flavonoids, sugars, quorum sensing mimics, and other hormonal compounds, which can influence the microbes that are residing plant environment. And in turn, microbes can also produce various chemicals which influence plant health. For example, auxin, if you have read basic biology, you will know that auxin is plant growth promoting hormone, right? But microbes can also produce auxins and influence plant growth. Apart from that, they can also produce several secondary metabolites, terpenoids, lactones, siderophores, and many others, which are quite important for plant growth and development. Okay, so with this knowledge, what I started to do is I tried to see what is the chemical signaling between the bacteria and the plants, and I focused on the rhizosphere bacteria, which are residing the plant roots, and how this chemical signaling and bacterial interactions influence plant health. And I focused on sulfur nutrition. That was a case that I took up. And so I asked the question how the interactions among rhizosphere bacterial members and their chemical signals, bacterial signals, impact plant sulfur nutrition. And why I focused on sulfur, you can see here, they can take up various oxidation states from -2 to +6, and they can be part of many essential metabolites inside the plants, right? Which is less in other major macronutrients that is required by plants. So with this, what I'll do is I'll ask the question and how I approached this problem, right? So the first thing that we wanted to know is whether plants' sulfur nutrition has any relationship with the microbiome that is in the rhizosphere. So that's my first approach to understand this interaction between plants to the microbiome. And then when I understood that, I took up the microbiome side and focused on how the microbes signal the plants and with a chemical approach. What I'm showing here on these blocks here are the approaches that I have taken. So first is I wanted to understand relationship between plant sulfur nutrition and bacterial function. So we have some resources where we can know how plant sulfur nutrition is affected in some of the genotypes or mutants. You will be familiar with mutants, maybe. And with these mutants, we first estimated their sulfate levels, which is a readout for sulfur nutrition. And we also estimated how the microbes changes in the rhizosphere. And to study microbes to plant, we developed a synthetic community where we bring the bacteria from the plant roots into culture, use them to develop communities, estimate their bacterial interactions, and study how it impacts plant sulfur nutrition, right? And I used metabolomics, some genomics to study the chemical signaling, right? So I'll be focusing on the first block here, plants to microbiome side, and how I did it, right? So I first screened for some Arabidopsis mutants from the literature, where we know that the sulfate levels may be different in those genotypes. So we took these genotypes. You don't have to read what are these genotypes, but we have our wild type, and we have genes for which sulfur uptake, transport and redistribution, and other regulation are mutated or disrupted, and try to see how the levels of sulfur are in those genotypes compared to the wild type no mutated plants, right? So this dotted line on the center shows you the levels in the wild type plants, and all these genotypes are being shown. I think it's a simple box plot being shown here. And you can see that we found two of the genotypes. The first two, the green MSA13 and HDI21. These two guys accumulated sulfur, which is higher compared to the wild type plants, right? So we focused on these two ones, which have accumulated higher sulfate and which have also accumulated lower sulfate compared to the wild type ones, and tried to see how overaccumulation of sulfate has changes in the microbiome function. So the technique that we use to understand microbial functions is called metagenomics. So we took these sulfur overaccumulating mutants and tried to see how the microbes in the rhizosphere changes in their metabolism, right? So MSA13 and SDA21, and these two are overaccumulating ones, as we have seen in the last slide. And col0 is the wild type guys, right? You can see that these overaccumulating guys have different levels of sulfur metabolism related genes. These are on the x-axis on the horizontal names. So we hypothesize that plant sulfur levels may correlate with bacterial sulfur metabolism in the rhizosphere. And bacterial genes related to sulfur metabolism, sulfur-containing amino acids such as cysteine or methionine, glutathione metabolism, and other sulfur metabolism-related genes are overrepresented or underrepresented, right? So this led us to the idea that sulfur metabolism of bacteria might be important for sulfur nutrition of plants, right? So in order to do so, what we did is we developed a synthetic bacterial community. If you haven't heard about this term, it's basically we cultured the bacteria from the plant roots. So we had a collection of bacteria from the rhizosphere region. We have it in culture in our lab. We know their genome sequence, and we know what are the functions they can perform. And based on those functions, we created a synthetic community composed of 18 members, which I named as SPAF18. And these 18 members are evolutionarily related with each other. And this represents a higher number of sulfur metabolic genes compared to others. So if you see here, I have simulated on the y-axis the number of random symptoms, and on the x-axis is the number of sulfur metabolic genes. My community, which is FAF18, lies on the very high range, which is around 170 or 180, right? So compared to other communities that we can develop from our collection, this community, which is 18-member community, is composed of very high number of sulfur metabolic genes. So that's why we call them functionally representative bacterial community. With this, what we did is we studied how these impact plant growth under sulfur deficiency, right? In sulfur deficient condition, you can see that when we do not apply the bacterial community, which is heat killed, which means it is inactive, it cannot perform, you can see a very drastic reduction in plant growth. And you can see when we give the active community, which is we do not heat kill, we keep them active and inoculate in the plant roots, you can see their growth is improved. This happens in sulfur deficient condition, but does not happen in sulfur sufficient condition when everything is normal, which means that only under sulfur deficient conditions, our SPAF18 community or synthetic community improves plant biomass, right? Which does not happen in sulfur sufficient or normal condition, right? So this has a beneficial effect to the plants, which is specific to plant sulfur deficient condition. And we also tested whether this beneficial effect is lost in this community when we develop a community which harbor less number of sulfur metabolic genes. So we developed a depleted syncom, we will call, and this depleted syncom has 40% less sulfur metabolic genes compared to the functionally representative SPAF18 that we developed. And what we saw is that the functionally representative community can improve plant growth, but the depleted syncom doesn't improve plant growth, which means that this functional role of sulfur metabolism is important for this beneficial effect of the microbes, right? So we wanted to ask, how does it do it? How does the community improve plant growth? So what we did is we use the community, which are active cells, so bacterial cells inoculated to the plants, and we also created cell-free extract, which we mean, but what we mean by that is we grew the bacteria and we took the exudates from the bacteria and we passed it through a 0.2 micron membrane so that no bacterial cells are present, only the secreted chemicals are present. And we saw a very similar increase in plant growth when we use the cell-free extract or we use them in direct contact with the plants. So which means that it improves plant growth via secreted chemicals. And then we hypothesized that this could be one of the sulfur-containing metabolite. So we screened some of the well-known sulfur-containing metabolites from our chemical library, and we found that glutathione, which is abbreviated as GSH here, gives the most highest response compared to others. So we hypothesized, okay, this might be the molecule that is present in the secreted chemicals in the bacteria, which is important for plant growth. So we deleted the glutathione biosynthesis gene in one of the bacteria, which is 6A2 here. You can see the effect that if we delete the glutathione biosynthesis gene in that bacteria, we see a drastic reduction in plant growth, right? Which means this glutathione biosynthesis gene is necessary for this beneficial effect that we observed in the previous slide. What we wanted to do further is to ask how the pairwise bacterial interactions within this community improve plant growth under sulfur deficiency. So we took a computational modeling approach from the genome scale metabolic models. I'll not go into the details. So basically what I'll try to explain is that we calculated the interaction score between the bacterial members and tried to correlate with the plant growth. So how did we calculate the interaction scores? We took two bacteria, we grow them in co-culture, A plus B, or we grew them in monoculture, where no other bacteria is present, and compared their growth rate. And we take the log ratio of that, which became our interaction score here. So when we calculated the bacterial interactions, what we found is that majority are competing with each other. You can see that because in general, there is scarcity of resources, which is what we observed. And if we put two bacteria together, they try to compete with each other. And we could estimate their interactions based on this computational approach as well. So what we further did is took the most competing bacterial strands pairs and the least competing bacterial strand pairs, 10-10 each, and tried to see how these bacterial pairs improve plant growth under sulfur deficiency. And we found a very negative, strong negative correlation between the plant benefit on the y-axis and the competition strength on the x-axis. So if competition between bacteria increases, the plant benefits more, which gave us the idea that there might be a fitness trade-off between plants and the microbes. So what we show here that when there is more stronger competition as indicated by the negative sum of the interaction scores on the x-axis, you can see more glutathione is released, and this glutathione improves plant growth. So when there is low bacterial competition, they release less glutathione, and when there is very strong bacterial interaction, they release more glutathione, and as a result, the plant benefits under sulfur deficiency. But as we all know, if two bacteria compete with each other, they do lose their fitness. And due to this fitness reduction, the plant's fitness is increased due to this glutathione release, right? So which we termed as a plant microfitness trade-off. And in essence, plant fitness is enhanced at the expense of bacterial fitness, which is what we discovered. So I'll not go into detail, and we'll delve into discussion. If you're interested, this study is now published. And if you want to read more, you can always scan the QR. Right. We can go for questions that you may have.
Thank you, sir. That is truly informative discussion. And we have now 5 minutes for quick question. So anyone
Yeah.
who would like to ask something, they can unmute yourself. I think there is so many hand raised.
All
Yes,
right.
Sampurna, yes, please unmute yourself.
Yeah. Hi, Dr. Arijit. It was really a nice presentation, really nice. I had one question. You said that you are using the highest sulfur metabolite producing bacteria or something like. So, did you use these kind of bacteria in plants which do not require or require less sulfite or sulfate for their growth? And did you see any difference in expression?
Yeah. So what we do is we use the model plant Arabidopsis, which is the equivalent to mouse in biomedical science. So
Yeah.
this is belonging to Brassicaceae, and they require a lot of sulfur. So our experiments were done in high sulfur requiring crop. We did also showed this effect, which I did not show today, in leafy vegetable, but that is also high sulfur requiring. We did not test in less sulfur recording crops. We do not know the
Okay.
answer.
So, okay, thank you. So, as a hypothesis, what might happen if this bacteria is used in a low sulfate requiring plant?
So the environment that we used is less sulfur levels in the soil. So even if you
Okay.
grow less
Okay.
sulfur requiring crop, if the soil is sulfur deficient, we expect that it might work. We are doing some testing and taking it to
Mhm.
trials. So those are different levels.
Okay. Thank you.
Thanks for the question. Yeah.